The ancient Greeks told a story about Prometheus, whose liver was eaten by an eagle every day only to regrow overnight. This wasn't just a myth, it was a startlingly accurate observation of a biological superpower. When a surgeon removes a piece of your liver, say for a transplant donation, they are initiating one of the most efficient construction projects in nature. The remaining tissue doesn't just get a little bigger, it senses a profound loss. Within minutes, the body triggers a cascade of events, a silent, microscopic alarm that screams for regeneration. This isn't scarring or patching a hole, it's a complete restoration of mass and function, a process so precise it feels almost intelligent. This regeneration isn't magic. It's a conversation conducted in the language of proteins. The primary signal comes from a molecule called hepatocyte growth factor, or HGF. It's not just floating around waiting. It's activated by enzymes that are released due to the injury itself. Think of it as a key that's been broken in half, and the injury provides the welder to fuse it back together. Once active, HGF binds to specific receptors on the surface of liver cells, the hepatocytes, like a key fitting into a lock. This single event unlocks the cell's internal machinery, telling it to prepare for a monumental task: division. Your liver cells are typically resting. They are in a quiescent state of the cell cycle known as G0, performing their daily duties of detoxification and metabolism without any thought of multiplying. The signal from HGF and other factors like TGF-alpha essentially kicks them out of this retirement. The cell enters the G1 phase, where it begins to grow in size and synthesize the proteins needed for DNA replication. This is a point of no return. Once committed, the cell meticulously duplicates its entire genome during the S phase, ensuring each daughter cell gets a perfect copy. All of this happens in millions of cells at once, a coordinated, silent symphony of growth. But the hepatocytes don't work alone. The liver is a complex city, and its regeneration requires all of its citizens. The non-parenchymal cells, like Kupffer cells, which are the liver's resident immune cells, become hyperactive. They clear out debris from dead and damaged cells, essentially acting as a cleanup crew that prepares the construction site. Simultaneously, sinusoidal endothelial cells, which form the lining of the liver's intricate blood vessels, begin to proliferate to ensure the new tissue has a blood supply. They lay down new capillaries, providing the oxygen and nutrients the rapidly dividing hepatocytes desperately need. How does this massive burst of growth not turn into cancer? The liver has an incredible sense of proportion. It doesn't just grow, it grows to restore the optimal liver-to-body mass ratio, which is about 2.5%. The secret lies in a balance of signals. As the liver grows, inhibitory signals, most notably a protein called transforming growth factor beta, TGF, begin to rise. This molecule is the stop signal. It binds to hepatocytes and pushes them back into their resting G0 state. The extracellular matrix, the physical scaffolding between cells, also plays a role, providing structural cues that signal when the architecture is complete. What's truly remarkable is that this whole process is mostly handled by mature, specialized cells. It's not an army of stem cells. The existing, working hepatocytes are the ones that divide. This is called compensatory hyperplasia. However, the liver does have a backup plan. In cases of severe, chronic damage where the hepatocytes themselves are too sick to divide, a small population of hepatic progenitor cells, sometimes called oval cells, can be activated. These are more like traditional stem cells and can differentiate into new hepatocytes and bile duct cells, acting as a reserve force when the primary army is overwhelmed. This dual system provides a level of resilience that is almost unique among human organs.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.